So Fundus had a Northeast challenge, a West Coast challenge, and I would say the person who just has been the heart and soul of putting together the Southeast challenge is Alicia Milliken, who is just an incredible woman, like really just a powerhouse and so brilliant and so lovely. And I got to finally meet her in person last summer, which was a big highlight for me when I went to NAMA. So I'll tell you about Alicia first. She's from Ohio originally, and really has a passion for the outdoors. She's a master gardener, and she is I think you were the founder and the president of the Alabama Mushroom Society,
Not
right?
the founder.
Not the
Not the
founder.
founder.
Okay, sorry. Okay,
No.
I don't want to say that right there, but really like the president of the Alabama Mushroom Society. And I mean, I have she did so much incredible work with this society. Like I've showed multiple people on our board, like, look at the newsletters they're putting out, like, look at this incredible stuff that they're doing. Really just amazing work. And she's now is just doing it is so powerful, doing so many amazing things, also on the executive board of NAMA. Also recently became a curator for fungi at the University of Alabama, West Alabama Herbarium, which is just like unbelievable, while managing all these things and while raising two beautiful children. So dang girl, like that's just so impressive. And anyway, Alicia is a really brilliant identifier, like a really dedicated scientist who, I mean, I can't even think of how many samples you have single-handedly like elicited, processed, gotten sequenced, and gotten deposited into various herbaria. So really just a cool person to know. And Alicia really, I think, had this idea for the Southeast Challenge, and a bunch of us were lucky enough to be brought in on it. And two of our other speakers not only helped in big part getting the Southeast Challenge going, but also bringing in this whole other dimension, which is using art really as a medium to convey science. And I think you'll be really excited to see how their work has impacted the challenge. So I'll tell you first a little bit about Patricia Larkin. So Patty, you'll see she'll be on, and she's wearing a fabulous shirt that she designed with one of her watercolors from the challenge. But she, is another great identifier, great citizen scientist, also an art teacher and an artist, really incredible artist. And her art is really focused on nature. And she does incredible work with items that she finds in nature. Like if you've seen, she does incredible paintings on skulls that she's found, and paintings of mushrooms. And anyway, so she has been working also with Fundis, not just to paint the species in several of their challenges, but also to do some fundraising by creating some sticker art, which I think I linked to in the email. But if anyone, I can also try to throw it in the chat too, because it's beautiful art.
I'm on it.
Oh, sorry. Yeah. Thank you so much. And then Amos Zeller, I hope I said his last name correctly. If I didn't, you tell me, Amos. Is another incredible person who comes to us from the Kentucky Mushroom Club. And sorry, I'm just pulling up. I just realized I need you pull up his bio. Bad job, Annie. Sorry, Amos, for the delay. But anyway, so yes, Amos basically has been part of the Bluegrass Mycological Society. He is one of the leaders of that group, does incredible mycology work for them. He has background in chemistry that he uses to explain the ecology of fungi. And I know their club has done really cool work, and I think, maybe had some neat events this spring as well. He works at Bellarmine University, managing their science labs and all their instruments and stock rooms, and does a lot of chemistry as well. So he synthesizes pigments and aromatics and cooks and draws. And I just think like the coolest thing to me about this group is not just the level of like really passion and motivation to do work really for only out of love and only out of curiosity, but also just the incredible multi-talented nature of this group. So we have anyway, really cool folks tonight, and I'll turn it over. I don't know if Alicia, if you want to take over, but really excited to have the three of them. So welcome.
Yeah, thank you. So what I'm going to do, guys, is I'm going to start my presentation, which I'll begin by kind of explaining about Fundus and what Fundus does, and then Patty, when we start talking about, let me see, am I sharing that properly? Yes. When I start talking about the Northeast Challenge, I'll let you come in and share your art and your stickers from the Southeast Challenge. And then Amos will have you come in and do your stuff, okay? And then we'll resume my presentation going into the deep dive of the Southeast stuff. Can everybody hear me okay?
Sounds great. And the slides look great. Thank you.
All right. Okay. So Annie did an awesome introduction for me. For those of you who don't know me, I'm Alicia Milliken. I got my start in 2020 with the Alabama Mushroom Society, which was completely serendipitous and a total whim. I moved to a new state and didn't know anybody, and saw a advertisement for a mushroom society meeting, and I was like, well, that's interesting, and took my 3-month-old to this meeting, and I got hooked, and the way mushrooms do just got sucked in. 6 months later, I was writing the newsletter, and a year later, I was the vice president, and a few months after that, I was the president. Very quickly jumped on board, started working with the North American Mycological Association, working on several of their committees, and then had a wonderful, amazing opportunity to get to start working with the University of West Alabama, and they were kind enough to make me assistant curator of fungi. So I am the sole person that handles all the fungal specimens, not lichens, but macrofungi, microfungi, all go through me for the herbarium down there. And of course, as pertains to our presentation tonight, I am the volunteer coordinator for the Fungal Diversity Survey. So, oh, you know what? Okay, so Fungal Diversity Survey, or FUNDIS for short, is the only nonprofit focusing on North American fungal diversity and conservation. Their mission is to protect biodiversity through conservation of fungi and their habitats by increasing knowledge and public awareness of their diversity and distribution, equipping and engaging community scientists, and partnering with land managers, conservationists, and scientists. Fungi play a vital role in the environment. They break down debris, they provide a network of nutrient exchange in our forests, they are a food source for many animals and certain insects. And certain insects' complete survival depend on them. 90% of land plants form mycorrhizal relationships with fungi. Some plants can't survive at all without these associations, and most decline significantly without them. We are losing fungi faster than we can document their existence, mostly due to habitat loss. The Global Red List, which lists threatened and endangered species, has only assessed 0.02% of total fungal species due to lack of data. For comparison, 4.03% of total vascular plants are protected. That's not ones that have just been assessed. They've way more than that have been assessed. These are ones that have been protected. So huge discrepancy there. So knowing which mushrooms are where and when is vitally important to being able to study and protect them, because we cannot protect what we don't know is there. So the Rare Fungi Challenge was an initiative started in 2020 to document rare, underdocumented, or potentially threatened species of fungi in North America. The first challenge launched was the West Coast Challenge, and it was very successful. The results after the first year included major range extensions of 2 of the target fungi and documentation of 7 of the 10 initially targeted fungi, resulting in 20 vouchered collections for further study. The next one was the Northeast Fungi Challenge, and then ours is the newest one. So the Southeast Challenge covers a little bit wider of an area than what most people kind of limit to the Southeast. It's due to shared ecology in the region. So all the states included are Texas, Oklahoma, Arkansas, Louisiana, Mississippi, Kentucky, Tennessee, Alabama, Georgia, Florida, West Virginia, Maryland, Delaware, Washington, D.C., Virginia, North Carolina, and South Carolina. And you can see here, this is a big area, but when we started looking at the distribution of a lot of these fungi, they kind of they cover this area. So we didn't want to limit and you know, cut out some states where these fungi might be popping up when they're equally rare there. And right now there's not a Midwest challenge. So we included as many states as we thought was appropriate. So the Southeast Challenge includes 20 species. They were chosen by a team of mycologists and experts for being rare, underdocumented, or potentially threatened. They were carefully chosen to cover a wide variety of morphologies, habitats, seasonalities, and all are easy to ID in the field. Our target species all occur within the Southeast, some widely, and some are only known from a very specific locale. For each species, I've provided an example photo and a list of the season, habitat, and identification features. All of these features should be considered when you're trying to make an ID, but when one feature holds more weight than the others, I've highlighted that key feature in yellow. So before we jump in, Amos, do you want to hop on and share some of the art that you've been doing and how you've been kind of talk about how you've been promoting the challenge?
So I hadn't ever drawn a mushroom before I was asked to do this, but I think it has turned out okay. You all can tell me if you have or haven't. But I've covered 7 so far, and you'll see a new one tonight. This first one here is Boletus atkinsonianus. And there's a picture of a pencil included for scale if you wanted to know how large they are. I've also got them all here next to me in a stack. This is Hygrophorus kokori. There we go. Kokorii. I forgot exactly how to say it. A really odd looking one with quite a bit of color to it. A tooth fungus, Hydnellum geogenium. This is the pawpaw polypore Philoporus amplectans. These are Wolfina aurantiopsis, a cup fungus. There's Lactarius purpureo echinatus. I think I got that right.
A fantastic job of exhibiting the latex on that. That is wonderful.
That's the one time I used white paint. Everything else is colored pencil, if I didn't mention that yet. And it helped out a lot there. And I don't believe you all have seen this one yet. This is Pluteus mammillatus. I just finished it last week and just finished making a little edit of it. If you're curious how these images came to be, each of them, I'll just show you all. You'll notice, especially on this one, things are not placed exactly where they are on the original drawing. But all I do is take a photo of them under a good lamp and move things around a little bit where convenient and kind of get rid of the stray marks that are on the paper. So what you see is really art. And yeah, I think I actually want to follow with Patricia's lead, Patty, and make some kind of merchandisable sticker or art for them, as long as people are willing to buy them. And
can you go back to the Pluteus? That's just so beautiful. Oh my gosh.
Yeah, so totally.
I'm so excited that we get to see the brand new one. Does anyone, just as like a audience engagement moment, does anyone want to shout out what feature of this Pluteus makes it really unusual? You don't have to know the species. You can just look at the picture because it's highlighted so well.
I'm not going to spoil it.
Yeah, don't spoil it. Literally anyone. Or you can put it in the chat if you want. Yeah, Elizabeth said in the chat. Yeah, Pluteus doesn't usually have a ring. Totally.
Mm-hmm.
Like, yeah, awesome. Like, and it's one of the, like, yeah, exactly. Oh, it's also a low ring, April said. Yes, that's so
really
true.
Low, yeah.
Yeah, Pluteus having free gills and no ring is like a pretty unique situation. Having free gills and a ring is much less unique. So this is interesting. Anyway, this is gorgeous. Amos.
Thank you,
Wow.
thank you, thank you. Yeah. I think it's the only one in our entire region that has any kind of ring on it. And the only other species I know of is also yellow and occurs in Europe. So it might be kind of like a counterpart to ours. I forget the name exactly. But yeah, if you are kind of curious about seeing any kind of like more in-depth information on these fungi, and I'm sure Alicia has some things in our project files, some public and some not yet, that have official descriptions. But I've been posting them on my personal Facebook with like long, almost like Michael Quo length or longer descriptions of their features and where they're found, if anybody wants to look at them. Patty also has really good descriptions on hers, and she'll talk about them, I'm sure, that are a bit more well organized. But yeah, if anybody wants to see any of them again or ask any questions about them, that's all I have to say, really.
Stunning. Thank you.
Okay, cool.
All right. Thank you so much, Amos. Really adding so much to our challenge and helping get the word out about what we're trying to find and what they look like by sharing this art. It looks like Patty is here and available now.
Hey guys.
Hi, Patty.
Yeah, sorry about that. I'm here now. I know I came right in at 7:45. I was like, oh my gosh, this went so much faster than I thought, and they're already this far ahead, and I didn't expect that, but I'm happy to be here tonight. And I want to say I'm not the first person who started doing stickers for Fundis. I don't know if everybody's aware, but I think it's called Califungi out there. I think that's what it is, but the Fundis group out in California was actually the first one to start it. So I was approached by the Fundis team and they asked me, they're like, hey, we see you have some interest. I had a couple finds of my own. I found Boletus purpurobellis. I don't know that I said that right. I just kind of stutter through that. But it was my first find, and Annie was actually the one that identified it for me. She's like, do you know what you have? And I was like, no, some red boletes, they're cool. And she's like, you have something really special. And it was like an exciting day to find that. So I had found that, and I also found Antiloma indigoferum on my birthday, which was super exciting, really close to there. Same thing, Annie was like, Patty, yeah, you've got another.
But
so
you should see
after
the environment that you found. I mean, you have such a special, your backyard is such a special environment.
It's so the New Jersey pine barrens are like a magical, mystical wonderland. And those two finds were like across like a main street from each other. Like one was on one side of the road and the other one was like a little further in the other direction. It was Maricel Patino found the Boletus purpurobellis. I'm going to keep saying that because it's so fun. She found that near to there as well. So it was like, it's just the magical, mystical wonderland. But we can't find, I'm going to stutter through this one too. Echinodontium balui. There we are. We're still trying. It was discovered in Forked River, New Jersey, and we're actually doing an expedition we got permission from a local refuge right by where it was found. It took forever to get permission, and we're going to go hunt for it actually this weekend. We're going to do our first hunt. I know. I don't know that we're gonna find it. It's kind of
road trip.
Like, if you can look it up for this Saturday, yeah, and we're gonna try again, regardless in late fall because it's gonna be better viewing. I know I posted it on my Facebook, but it was a while ago. I should have thought to like be like, but if you can make it up this weekend, seriously, we'll get you in there. I'll get you in. So we have our consent form, so we're gonna hunt for that one this weekend because that one hasn't been spotted here since it was discovered in 1908, I think 1908. But it's been spotted elsewhere, and it's been spotted by I can't remember who it was.
Bill Neal.
Who was it?
Bill Neal.
That's it. You knew it. Yep. So, and he's given me some good tips as well. So we have some good tips. And then one of the people that works at the refuge actually is like, don't check that section of forest. That's going to be a waste of your time. Go check this section of forest that has older white cedars. And we're like, okay, we're going to go do that then. That's what's going to happen. Yep. So we'll see. Yep. So we did some sticker sheets for ours, which were fun. We have 3 different sticker sheets. And I've also been doing taking the illustrations that I made on watercolor paper and then adding them onto other things so that they're like actual art pieces that can also be purchased for Fundis as well. All of these are for a
Wow.
Fundis fundraiser. And
You have an auction. There should be like a
what's that?
there should be like a Fundus online auction for this stuff because I feel like people, different clubs would want to get in on it. That's so cool.
Yeah, I do know, and I can say that too because I don't know that Mandy knew that part of it. I'm not sure. I think she's just like, okay, this is happening and that's happening, and I have the Fundus team, but I don't know if they're all in on all the aspects of it. I'm not sure. We'll make it more cohesive. But something that's going to be coming down the line will be t-shirts and sweatshirts as well.
Very
That was
cool.
a big long ramble. Does anybody have any questions? Did it glitch?
All right. Thank you, Patty. If anybody has any questions that they think of, they can drop them in the chat and we can come back around. But we'll go ahead and dive into the species that we're going to be looking for with the Southeast Challenge. All right. So the first species in our challenge is Winia spracioides, the stalked cauliflower fungus. This mushroom fruits from early spring to late fall. It grows in soil and is often hiding under leaf litter. It fruits from a dense mass of mycelium called a sclerotium. If you find this one, please cut it at soil level and leave the underground sclerotium alone so that it will fruit again. This is one of the few fungi that shouldn't be dug completely up. They are documented in the northern range of our target area in Maryland, West Virginia, Virginia, and North Carolina. And we actually just recently received a specimen collected from southern Mississippi, but unfortunately it was not dried sufficiently. And it was completely overtaken by mold. So we're going to keep an eye on that spot and hopefully find another one. And you can see there's a big note here that if you apply KOH to the surface, it turns magenta to bright red. Our second fungus is actually a whole genus, Glowiomucro, the glue drips. These transparent yellow-green spikes fruit throughout the year when conditions are favorable. Being a jelly consistency, they favor wet and humid environments. They fruit on decaying logs that no longer have their bark. They have been documented across a wide range of our target area, from Maryland down to Florida and west to Mississippi. If you find these, remove them from the wood and place them in a pile on a plastic bag or a clean plastic lid before drying, because they dry up to almost nothing. They look like a more my note doesn't make sense. The only look-alike are these more opaque Mucronella species. You can see these are green and much more transparent, and the Mucronella are much more opaque. This next fungus isn't much to look at, but it has the best texture, Rhytoporio astrosinensis, the bubblegum polypore. It stretches just like bubblegum. This is a crust that grows in and on downed and decaying pines that have already lost their bark. If you break soft wood from a decaying pine, like you see in this bottom photo here, and you see this white stretchy interior, you found the bubblegum polypore. When fruiting, it forms a crust with irregular pores that will be, they will also stretch like bubblegum when they're fresh, but the crust itself may not necessarily be present. You can find this one year round, and it has been documented across much of our target area, from Maryland down to Florida and west all the way to Texas. Next, we have Wolfina aurantiopsis, the lemon buttercup. This is the summer a summer fruiting fungus that grows from dead twigs and logs of hardwood trees. It is known to occur across our range from Delaware down to Alabama and west to Texas. They look a lot like the more common Galliella rufa, but they have a more yellow center, and the big tell is when you cut them in half, they have this opaque white interior, whereas Galliella is clear when you cut it in half. If anybody has any questions specific to these fungi, go ahead and chime in. I'll try and catch them. Otherwise, we can catch any more broad questions at the end. Multifurca ochre compacta, the fork gill. This mushroom doesn't look like much with its cream to buff colored cap, but flipping it over, you will find yellow-orange gills that fork repeatedly and smell like lemon when they're fresh. You can tell this one from similar members in the same family by the many forking gills that do not ooze when cut. These do not exhibit latex. It's been documented from West Virginia down to Georgia and west to Texas. This is the second least documented fungus in our challenge, Russula lilacipes, the purple foot russula. This is an unassuming mushroom until you dig it up and discover its lilac purple base. Keep a special lookout for this one in August and September in Virginia, West Virginia, and North Carolina. It's only been collected 16 times from 7 locations, and 10 of those collections come from a location at Arlington Cemetery in Virginia that is now paved over. That's correct, right, Annie? I believe you were the one that went and looked.
Unfortunately, yeah.
So that most prolific location for these is no longer viable, unfortunately. We actually just had a collection made in Virginia a couple months ago. It wasn't even in August and September. It was a totally new area in somebody's yard, unfortunately, which means we can't protect it or, keep an eye on that spot. And she didn't even know what she had. She posted it to a Facebook group asking, and one of our team members saw it and spotted it. And luckily we did get a sample of it, and part of it has gone to Dr. Bark Buke in France, who is the Russula expert. So we're hoping to get some sequencing done on it because this is from a very old lineage of Russula. It's one of the fetid Russula. You can see the top is that brown, dull kind of. This would be one that I would probably skip over in the woods if I didn't know to specifically be looking for it. And the new collection that was just made was over 100 miles south of the only other known location in Virginia. So I'm hopeful that we may find some more if we let people know to be looking for it. This is the first bolete on our list, Boletus atkinsonianus, the pastel bolete. This bolete grows from soil in mixed woods under oaks during the summer. It has a red to cinnamon colored slimy cap and white to yellow pores. It often has ridges running up and down the stem, and the stem will bend before breaking, much like asparagus does. It can look similar to Aurora boletus species, but they taste sour when you lick the cap, while this one tastes mild to sweet. It also has slight pinkish staining inside the cap when cut in half from top to bottom. It's been documented in West Virginia, the Carolinas, and Georgia, but also all the way over in Texas. So it possibly occurs everywhere in between, and we're hoping to find out. Hygrophorus coceri. This is Coker's wax cap. It's a small brown mushroom, only 1.5 to 7.5 centimeters tall. Could be easily confused with a number of the other small brown species at a glance. Look for a depressed center with the gills at a wide angle from the stem. Although that's not always the case, if you find a group of them, there will be some that exhibit this feature. The cap has fibers radiating from the center, and the stem is hollow. They also have a white spore print, which will help separate them from many of the other small brown mushrooms. I actually have an exciting story about these ones. Holly Keating and Stephen Lansadell were up visiting Alabama from Florida, and we all wound up in the woods together, and my kids were being awful and started having meltdowns. And so me and Holly, she was helping me get the kids to the car, and we didn't take the path. Of course, we take the way through the woods and are sliding down this hill, and I'm a little bit ahead with my kids, and she's coming behind, and she scoots through these brown mushrooms and grabs one to show me because she knows that I'll be excited to see some mushrooms because there wasn't very much fruiting. And we get to the bottom of the hill, and she's like, "Here, look." She was not prepared for me to be as excited as I was, because what she handed me was Hygrophorus kokiri. And how serendipitous that of all the people that could have been in the woods with her that day, it was myself, a member of the Southeast Challenge, who was familiar with this species. I forget. It's only like the 7th time it's been documented ever. And I found a whole big patch of it. We were able to climb up on the back up the hill and make some collections and some excellent documentation, and we'll be able to get it vouchered and sequenced. So it was very exciting and totally unexpected. Lactarius purpureo echinaceus is the purple spiny milk cap. This gorgeous mushroom occurs in the summer, growing from soil in mixed woods. It's been documented in Virginia, Tennessee, North Carolina, and Georgia. You'll recognize it by the purple fibers on the cap that form a radiating pattern around a depressed center. The gills, when cut, ooze white latex that does not stain the gills nor change color when it dries. We found this at NAMA last year. We believe we found this at NAMA last year. There's some funny things with Lactarius as far as sequencing, but we're working on getting the type sequenced so we can confirm that the ones that we sequenced are indeed Lactarius purpureo echinaceus. I'm optimistic that they are. Phylloporia amplitans, the pawpaw bracket. It's easily ID'd by its occurrence on living pawpaw trees. Most polypores grow on deadwood or up from the ground. These are soft, finely velvety brackets that often entirely encompass small branches of pawpaw. It's the most common fungus on our challenge list and occurs in decent numbers in the areas where it grows, but its host tree grows much further north than this polypore is recorded, so we're looking at better understanding its distribution and growth requirements. Anytime you have a fungus that relies on a very specific species like this one does, it could have a conservation concern. This should look familiar to you from Amos's beautiful artwork. This bright yellow wood grower is Pluteus mammillatus, the ringed shield. You'll find these from summer to fall, and they've been recorded from Virginia, the Carolinas, Florida, and Mississippi. Although there are several very similar yellow mushrooms that grow on wood, this is the only one that has a ring on the stem. But like was pointed out, you need to look for it really far down low on the stem, not far from where it attaches from the wood. It would be easy to miss if, some of these wood growers, they're stemming from further down in the wood, and it can be kind of hard to see the complete base. And this is something that could be missed in photos if you aren't really looking for it. If you're just getting pictures of how it's popping up from the wood and not fully excavating it, that lower ring could be missed. But if you see it, it's a surety that this is what you have. The frilly rosette, Podocypha aculeata, can be found growing from soil under hardwoods from fall to early winter. It has tough, leathery fronds that are frilly and reminiscent of a ruff leaf fabric. They start out white, but they may turn brownish, grayish, or pinkish with age. It's been reported in Tennessee, the Carolinas, Alabama, Arkansas, and Mississippi. Hydnellum geogenium, the yellow copse hydnel. Look for this mushroom from July through September, growing in duff under spruce and under conifers. It's a tough mushroom that is bright yellow and develops a dark center in maturity. The underside has bright yellow spines and often has multiple stems. It's only been recorded in Tennessee, the Carolinas, and Georgia. The ogre polypore is the perfect name for this big, ugly mushroom. Polyporoletus sublividus grows from July through October from soil in mixed wood forests. It has a purplish olive green cap and pores on the underside that run down the stem. It's tough and corky, and the stem is large and short. At maturity, these are an impressive mushroom that would be very difficult not to marvel over. Take a look at that bottom photo. Sydney Grimm, a friend of mine, that her hand is all the way around that stem, giving you a pretty good indication how big these are. These are massive mushrooms. Mycena carolinensis, the little yellow bonnet. This might be a difficult to ID mushroom if you don't first consider what it's growing from. These grow from sticks of rhododendron, swamp dog laurel, and blueberry, which are all shrubby small trees. The caps are a dull yellow and in the shape of a lampshade. The cap may be slippery when wet, but the stems are not, and the gills do not run down the stem. This is another one that would be very easy to miss if you're traipsing through quickly and not stopping and looking closely. Ammonoboletus weberi is known as the decorated bolete. You'll find it growing under pines and oaks from late spring to early fall. The key features here are the red pores that do not change color when scratched or cut. The stem has dots and streaks of red toward the top of the yellow stem and olive brown dots on the lower half. Look for the cap to have cracks and fissures showing the yellow underneath. Amanita westii, West's Amanita. This mushroom looks a lot like Strobiliomyces, that is Old Man of the Woods, if you're just looking at the cap, especially when they're young. The large warts that darken with age, a large bulbous base, and gills that start white and turn brown. There's nothing else that is quite like this mushroom. You'll find it from summer to fall, growing from soil and mixed woods. It has so far only been recorded from the Gulf Coast states, Florida, Texas, Mississippi, and Alabama. Agrosipy andersonii, the sandy waxcap. This is an appropriate name because growing in sand dunes is the key feature to tell these red-orange mushrooms from all the other very similar soil-growing waxcaps. They will always be growing nearby Florida rosemary, which you can see down at the bottom there, and it's only recorded in Alabama, Mississippi, and Florida. Auroroboletus abruptobulbus, the sand bolete. Another one that grows in sand dunes. This one prefers older dunes with pines and oak nearby. Its Latin name is an homage to the abruptly bulbous base of the stem of this mushroom. Look for this one in the fall and winter. It's only known from Alabama and Florida. This gorgeous mushroom is Entoloma gainesvilliae, the blue clawed. This species name is due to this mushroom only being known from Gainesville, Florida. This is the rarest mushroom on our list with only 5 observations on record, 2 in June, 2 in July, and 1 in October. There isn't anything else like this mushroom. The bright blue cap and the pink gills define it. This one is definitely on my list. And actually, speaking to Steven Lansadell, he lives, I think, 100 miles south of Gainesville and has a spot where he has documented these before, and on my insistence, he posted them to iNaturalist so we could record it, and he's hoping to find it again this year. And Elizabeth, you need to go to your hometown and find some Entoloma gainesvilliae. So you found a species on our challenge list. Now what? We're going to post them to iNaturalist, which is a social network of naturalists, citizen scientists, and biologists built on the concept of mapping and sharing observations of biodiversity across the globe. Submit photos of what you find, and others will help you identify them. This data is utilized by many organizations that assemble data for conservation, researchers, and mycologists. Uploading your high-quality photos to iNaturalist will contribute to the data we have on what fungi occur where and when. iNaturalist is free and available as an app and on desktop. I'm realizing now, Annie, I have a whole program. I have the whole second part of this presentation walking through iNaturalist. Is that something you want me to go through, or we can kind of cut it?
No, that's great. I think it's always, I mean, we have some people who are avid iNat users, but some people could always use it, I think.
Okay. all right, we'll do it. So the next part of this presentation is intended to be interactive. So if you have never done iNaturalist before, I'm going to walk you through it and we're going to do it together. So go on your iPhone, find the iNaturalist app. You're looking for this green bird, not the green background with the leaf. You do not want Seek by iNaturalist, and go ahead and get that downloading, and we're going to do this together. While it downloads, go ahead and set up your free account, and when given the option, you want to allow location permissions for iNaturalist and for your camera, and you may have to access your settings to do this. While it's downloading, let's talk about some of the photos needed for making a high quality observation. These are the photos and the information that you should always be taking down. Take a photo of the area that the mushroom is growing before picking it. Show the substrate is growing on and the leaves on the ground to show what trees are nearby. Take a close-up photo of the cap. You'll notice that the cap is taking up the majority of this photo. We don't want to see a tiny quarter-sized mushroom cap with a big old background all around it. We want to be able to appreciate the texture. We want to be able to see it nice up and close and in focus. You're going to carefully dig the mushroom up, taking care to get the complete base. Take a photo of the underside showing the gills or the pores or the teeth. Take a photo showing the stem. Take your time, get them in focus. Take several photos if you need to. I never get good photos the first time. I'm taking 5 or 6 and choosing the best one from those. If the mushroom you've collected has an important feature noted in our presentation, like bruising when it's scratched or not bruising or latex, you're going to take a photo of that too. For some species, this information will determine if we're able to name your mushroom to species or just offer a vague ID. So these are some examples here. Place the mushroom in your collection container with a bit of the substrate. We recommend a plastic tackle box to keep mushrooms separate and keep them from bouncing around or smashing into each other and destroying fragile features. When you get home, place the mushroom in a food dehydrator with a label. If you're trying to dry multiple, I frequently and many times have thought, I'll recognize these once they're dry. That you can't. There are many, many mushrooms that look just unideable once they're dry, and you won't be able to tell that little brown one from this little brown one. So just take the time to label them. Dry them at 110 degrees Fahrenheit or less. Higher temps start to affect the DNA. Dry for 24 to 48 hours or longer until they are cracker dry. Any remaining moisture will cause them to mold, and then I'll be sad and announce in my presentation that my amazing collection that you sent me was moldy. While they're drying, you can upload your photos to iNaturalist. Dry mushrooms should be placed in a plastic bag and labeled with the iNaturalist number. So now we are going to work together and we are going to make an iNaturalist observation together. You can post a photo of a mushroom you've taken previously, or you can take a quick photo of your cat or your dog or an insect or a plant. It just has to be something living. So this is the screen that you're going to see when you open your iNaturalist app. On all of the following slides, Android is on the left and iPhone is on the right. To make a new observation, select the green plus sign on Android and the camera icon on iPhone. You can take photos directly from the iNat app, but they are much lower quality and I don't recommend it. Select the album your photos are stored in. For most of you, it should just be your camera roll. You can select all the photos you want to include in the observation from here. If anyone is actually following along and I'm going too quickly, please say something. On Android, you can click and drag the little photos at the top to reorder them. Then click on What did you see to enter your ID. It will automatically suggest IDs, but iNat AI is not any better at IDing mushrooms than any other app, which are all pretty bad. Many of the suggestions you see here in this screenshot are all wrong. I'm going to click the species search box to type in my ID. If you don't know what your mushroom is, if you cannot name it even to genus, just name it to fungi. People like me search iNaturalist and I'm searching for fungi. If you put it in that kingdom, people like me are going to see it and we're going to try and narrow it down for you. If that's all the best you can do is you say, this is a mushroom, that's good enough. Here's a hot tip. If you type in the genus name with a space after it, it's going to give you a list of species in that genus, but not the option to choose just the genus level ID. If you remove the space at the end, you can choose just the genus level ID. If you have no idea what it is, like I said, just type in fungi and it'll give you kingdom fungi. On Android, when you choose an ID, it will automatically pull up information about it, and you have to choose select to choose it. On iPhone, if you want to see the info about the species, you have to select the little I info button. To select the ID, just click it. After you've selected the ID, it will take you back to the observation, and you should type in any notes about what it was growing on, what kind of trees are nearby, and any other details that will be difficult or impossible to tell from a photo, such as the odor, the taste, the presence of latex, or the texture. Your location permissions are turned on your phone for both iNaturalist app and your camera. Your location should automatically fill in. Click in and confirm that it is correct. Where I live, it automatically thinks I'm miles away from where I actually am, so I always double check it. From this screen, you can click on projects if you would like to add your observation to a project that you have already joined. If you have not yet joined a project, you cannot add it. Some projects pull observations automatically. They are called collection and umbrella projects, and they'll be at the bottom of your list of projects. They cannot be selected. We'll talk about joining projects in a minute. When you have input all of the information you have and you chose any projects you want to add them to, you submit it by selecting the check mark on Android or the share button on iPhone. And you can see it's uploading. To join a new iNaturalist project from your main screen, on Android, select the 3 lines in the upper left corner of your screen. From the menu that appears, choose Projects. I didn't put, but on iPhone, you're just going to click the Projects button here on the bottom. This will show you the list of all the projects that you have joined. If you have not joined any, you're not going to see any listed here. To join a new one, choose the magnifying glass in the top right, type in and select the name of the project you want to join, and when it pops up, select it, and that's the case for Android and for iPhone. This button that is partially obscured says join, and once you click it, it will change to leave. Then just hit the back arrow to exit out of this screen. Finding your iNaturalist number. This is a unique ID number for each observation. On Android, choose the observation you need the INAT number for and scroll to the bottom. The INAT number is listed under ID under metadata. It's easy peasy. On iPhone, choose your observation you need the ID, the INAT number for. Click the share button that's shown here, circled in red. And this will bring up the link for this observation. Copy and paste the link somewhere into a note, a message, anywhere. The INAT number is the number after the backslash on the URL. You'll need this number if you're sending me a collection from the Southeast Rare Fungi Challenge, or if you're mailing a specimen in for DNA sequencing, or if a researcher has requested you send it. Having it linked to the iNaturalist observation so they can easily see all the data, the location, the photos, makes it extremely convenient. And people like me, I get hundreds of mushrooms sent to me a month. So we may have discussed it. If it's not labeled when it comes to me with an INAT number, I'm not going to remember what it is. You can find detailed instructions on this whole process and uploading to these websites from the QR code here. You can reach out to us at southeastrfc@gmail.com for ID confirmation and mailing instructions. All the collections of the target fungi will be sequenced and accessioned at the University of West Alabama Herbarium. And always be aware of the local laws regarding required permits for the land where you are collecting. Elizabeth reminds everyone that this is being recorded, so everybody can come back and find these details on YouTube. And that is it. Does anybody have any questions about any of the project or any of the mushrooms included? Melissa, yeah, Devin recently found Lactarius purpureo echinaceus too, which was really exciting. And again, waiting to be sequenced to confirm, which sequencing isn't as easy as just running it through a machine and it popping up and saying Purpurea echinaceus. Until we have the type specimen sequenced, which we are working on, we don't have a sequence to reference to be sure. And interesting about this species is the sequence indicates it belongs into a totally different section of Lactarius than is currently in, but none of this subsections, none of the sections of Lactarius are supported by sequencing. They are organized by macromorphology by previous old school mycologists. So sequencing all these types might throw a whole wrench in Lactarius taxonomy. We will see.
That would be some good trouble.
Yeah, well, I mean, it's only a matter of time. We need to get it all sorted out. We know that morphology isn't a great indicator of how mushrooms are related, so it's something for the mycologists to tackle, I think.
Well, I just think
Moni and
go ahead. Yeah.
Moni and Steve asked how long the challenge lasts. Not forever. We're going to run it for a couple of years and we will reevaluate the data and see if we need to continue, if we need to take some specimens off the list. We'll reevaluate and we don't know. It's going on until further notice.
So it sounds like a lot of your experience so far is just like these mushrooms are rare enough that it's exciting to find one at all, and you're excited to document them. But at the beginning, you talked also about sort of the link that this could have to conservation, and I'm wondering sort of, what are the next steps once Fundis has confirmed that a mushroom is somewhere? Like, what are you hoping to do with that information?
So, okay, so well, there's a lot that goes into it as far as data, like analyzing the data and figuring out. We have to basically make a case for it being a rare a rare mushroom, and then they coordinate with there's a conservation group that they coordinate with to try and get the mushrooms on the IUCN list. And then once it's truly rare, then it's a matter of laws and what, when something is listed on the IUCN list as rare, then it's a matter of the hoops that a new shopping mall would have to go through to pave over that area, and et cetera, et cetera.
So is the theory sort of you've put together this list, all the mushroom clubs are looking for these mushrooms, and if after the 5 years that everyone has been looking for these things, you've only found a few specimens, then that's the argument that they're very rare mushrooms? Is that sort of
and right, what we're trying to determine, so there's a bunch of us, Devin Merkel is in this call as well. There's a bunch of us that are amateurs, but we've also been working with Arlene and Alan Bassett and Bart Buke and many mycologists who have contributed to this list. So what we need to figure out is, we know that these mushrooms haven't been recorded very many times. And is it just because nobody's really looking for them, or because they're truly rare? And that's the data that we're looking for. And then once we get more data, even if so we've only found it so many times, what is the common denominator? Are we seeing consistencies in habitat? Is it only certain times of year? Is it only when we've had a lot of rain? Like, we're trying to find it more to get more data so we can figure out what needs to be protected. Many of these have only been found so few times that we really don't know variability in their habitat or variability in the season, or they're just, everything is so data deficient. We don't know what needs to be done with it.
The
Did that answer
Southeast
your question?
the Southeast is probably even less documented than the Northeast in some ways. I mean, like, I'm just thinking for Patty's area, like the Atlantic white cedar is already like that kind of habitat, like Atlantic white cedar swamp. It's like an extremely threatened habitat. Like it was very prevalent, and then people just ripped it up to make boats and stuff like that. And like at least 3 of the mushrooms on the Northeast Challenge are from Atlantic white cedar swamp habitat. And in New Jersey, like there are definitely a lot of conservation efforts to try to create new swamps, like there are new plantations, obviously to protect old swamps. And some of these, like the mushroom that Patty was talking about, the kind of Dontium bluei, like it's not even going to grow on anything, any ones that are like less than how many years old, Patty? Like, I mean, what's the age?
45 years or more to grow, which is wild. So we're looking for old age or older growth Atlantic white cedar trees.
So I feel like in a lot of cases, like like if you do find something truly being rare, to me, like it's like a marker of a threatened habitat, um, you know, more than anything. And then it's like seems like a good I mean, obviously there are a lot of reasons that we should want to protect cedar swamps, but the fact that there are like species that will actually be lost if we don't have any cedar swamps is I think fairly compelling.
Yeah.
Add to it.
Yeah, there's and I mean I mean the fact that we have that data already is a huge boon, knowing that we're looking at trees that are, swamps that are older than 45 years that have been established that long.
Yeah, we don't know that about so many others. Like the Rushla that you were showing, Alicia, the Lilacipes, Rushla lilacipes, like that. It was described by I think it was described by somebody who was at the USDA when the USDA was still in Northern Virginia. It was where it was basically where the GW Parkway is now, and several of the areas where it was found in like the 1940s and 50s were areas where there was like a lot of parkland and even residential neighborhoods, but still with like trees and lawns. So I went just to walk the whole area to literally try to go to the places where this had been recorded, because Alicia was just collecting this incredible compendium of like information about sightings of these mushrooms. And if you go to that part of Northern Virginia, essentially it's not just like one small area that's paved. It's like Crystal City, it's like the entire area is gone. And so we can't even know what the trees were that it was associated with. Like, now at Arlington Cemetery, there's like a lot of ornamental trees, but you can look at them and eyeball them and know that they're not even 50 years old. A lot of them, they look pretty new. There might be some older ones, but so I just think like it's one of those things where like, so if well, we've already lost like this whole site where this mushroom was described, but then someone finds it in like a surprising part of Southwest Virginia, like if it wasn't on someone's lawn, maybe that could be, a place to protect. I don't know. I think there's the opportunity for these kinds of like, that's what I would hope would come. But
Yeah, right now it's really about data collection. What we desperately need is data on these mushrooms. They've been found so few times. We know so little about them, and so we don't really know, because there have been some that have been proposed on other challenges that turns out that they're not rare. Once we spread the word and we encourage our clubs to go out and start really looking for them, they popped up everywhere. They've just been sort of flying under the radar and sort of ignored and missed. And we know that we can focus our efforts elsewhere.
One really cool thing for our club is just the fact that we really are in like a weird location where we have species from the Southeast Challenge that should be here. We have species from the Northeast Challenge that should be here. And so it puts us in like a really unique position. And, like depending on the season, we'll find,
Right.
and depending on the microhabitat, we can really find different things. So it's really something kind of cool for us. We're lucky. We have like a weird, I don't know, a weird crossover ground.
And who doesn't love a scavenger hunt? This is the ultimate mushroom scavenger hunt. I dropped a link in the chat. Someone had asked about the poster, and it's available to download from the Fundus website. And I would be more than happy to send my presentation, all the slides, to anybody that wants them, or if the club has a website and wants to make them available there, that you can download them yourself. You can print them out and have them on cards and handy. Sort of has everything you need to know right at your fingertips that you want to have on small cards in your pack when you're in the woods, or whatever you have to do we printed out the big sheets and had them at NAMA last year, just to be a nice reminder when we're out on forays.
The poster could be something good for us to print too, to have at like our mushroom fair, for example, because when we do our mushroom fair, we have forays from all over the area, including from parts of Virginia and like, farther than we usually go. So it could be a cool time to have them all up there. So we look at the table and say like, hey, is there something on this table that we need to look twice at? Because a lot of these some of them are really eye-catching, but some of them are not as eye-catching.
Right.
Like the hygrophorus species that you found, like if you hadn't been there, yeah.
No, I mean it wasn't, it's a small brown mushroom. Prior to knowing about this species, I probably would have been like, that looks like something hard to ID, and I would have left it in the woods. Patty says she keeps the printout packet in her hiking backpack. Yeah, Devin has a great idea to consider asking local parks if you can print out the challenge poster and like post it to their map area at the trails. That's a brilliant idea.
It's literally like America's Most Wanted. Like this is like we're
right, yeah. Mushrooms Most Wanted. And like I dropped it in the chat, the link to the big poster, and then Annie, I'll send you my presentation and you can print the slides or have them available however you wish.
And we can include it in our newsletter as well if people want to check the slides. Yeah, that would be cool.
Awesome. Well, thank you so much for having me on, and for everybody taking a look at the challenge and considering going out and finding these some of these rare mushrooms.
Thank you so much, Alicia. I just have to say, like, there are so many people who have worked on this challenge, and Alicia is just like, other than being like a very brilliant and dynamic person, is like one of the most incredibly like motivating like leaders that I have ever seen, because she got literally, it's probably like herding cats to get like, how many different mycologists did you get to work on these descriptions? And then she managed all of us like amateur people who did a lot of other legwork. Like, how many people worked, like so many people, and she just kept the entire thing together and kept everyone on the ball. I mean, it's like the level of executive function and vision that this person has is like just phenomenal. I mean, it's like really unbelievable. So to me, it's like, these kinds of projects.
I just looked, there's 22 people, not counting the mycologists, but 22 amateurs on the project that have participated, but I couldn't do it. I wouldn't do it without all y'all's help.
Managing that many people is like literally so hard, and managing that many mycologists, because you also had Arlene and Alan, Bart, Jacob,
Jay Justice, Elise, Bart Buke.
Just really figuring out who all of the go-to people were, getting them all on the project. I mean, it's just really tremendous work. So I feel like I'm just so inspired, and I think it really tells you, like, we, as a club, like we could think of, like, I don't know, we don't have to make our own challenge, but we could, Elizabeth, like we could think of like, are there things we want to look for? We've talked in the past about trying to document all the species on a small area like Roosevelt Island. Like there's so many I mean, there's so many things that we could do, and so yeah, it's just really cool to see what one person can pull together with a vision and leadership. So
And
Yeah, and
motivation. You just
we
need to, you don't have to be an expert. You just have to be motivated. You just have to be motivated.
We have been trying to build relationships with some of the local parks because in a metro area like DC, foraging is pretty much completely outlawed in all of the parks, right? And so we're trying to like build relationships and get them to understand our interest in documenting the species that they have in their parks. And I love the idea of sharing that poster with them of like, you might have rare species here. Will you help us crowdsource finding them? As another little thing to like
it's
sort
a
of
great
show
idea,
them what
Elizabeth.
we're about.
That
Yeah.
is a really good idea. Like bring them into
Um,
the
my friend Jessica works for the Ohio Metro Parks up in Ohio, and they are trying to document all the rare species that they have in their metro parks so that if they decide to put in new facilities or something, they know that they're not bulldozing an area that ought to be protected within the metro park. And that's a good way to look at it. Like if you guys are looking to put in trails, you don't want to put trails in over this important species. But do you know if it's there? Like maybe we should have state parks, all of it, state parks in Alabama also outlaw foraging. It is illegal to forage in state parks in Alabama and Georgia, and the fines are hefty. So trying to go at it from a different angle and a conservation angle is a good way to get them involved.
Well, thank you again. This was really great. Thank you so much, Patty, Amos, Alicia, and thank you, Devin, and everyone else who is on and listening who's helped out with the challenge too. I mean, like people have done so much, and it's been really fun to just play a minor role of getting to be part of this cool group. And I really encourage everyone in our club to really latch on to some of these things like the DNA committee or different projects, because it's really neat to see what people can do when they come together. So anyway, thank you so much.
Yeah,
Thank
thanks
you.
everybody. Have a good night.